So this is an unusual talk for us in the sense that typically one of us goes somewhere and gives a talk and the other one stays home. I don't think we've ever tag teamed like this, but that's the plan for today, is that we're going to give about a 50-minute talk. I'll do the first half more focused on mechanisms. Edith will do the second half more focused on moderators and resilience, and we'll see how it works. I should speak for myself and not her in saying that if you want to interrupt with questions or comments or arguments, that's totally fine. I'm not wedded to getting through any particular agenda here, so I'm happy to interact. It's more interesting that way. As Paul said, half of our appointment is in the Institute for Policy Research. We're accustomed to economists, and there are different ways of interacting than psychologists and sociologists. So it's fine if you want to argue about my title slide, we can do that. If not, I'll go on. There's somewhat of a culture in our group of making fun of each other's disciplines. It's all in good nature, and we like hanging out. So let's see. Let's get started. So one of the things that has been clear for many decades, thanks to work done by people around here at Davis and elsewhere, is very clear evidence that early adversity in many forms, in terms of low socioeconomic status, maltreatment, harsh parenting, long-term separation from caregivers, is related down the line to adverse psychosocial, behavioral, educational, occupational outcomes. We know that kids who grow up in tough circumstances tend to struggle overall with doing well in school, with getting high-paying and good jobs. They have increased risks of many different psychiatric disorders and encounters with the criminal justice system. So none of this is news, right? But what has been news over the past decade or so is evidence that these effects go broader and deeper. So broader in the sense that they don't seem to be restricted to mental health, occupational or educational outcomes. There's now, I think, quite compelling evidence from studies that I'll talk about that they extend also to physical health, and specifically to chronic diseases associated with aging at the end of the lifespan. I'm talking about things like heart disease, some forms of cancer, autoimmune disease, and even premature mortality. And that's been an interesting epidemiological development that I think many people weren't anticipating. So I could show you lots of different figures to depict this relationship. Here's one that I think is particularly illustrative. It's a study of about 5,000 Britons who were picked up in a birth cohort study shortly after World War II, and at the point this analysis was done in the early 2000s, they were in their mid-50s. And what you can see here is that survival curves diverge when people are in their early 40s according to the socioeconomic status that the cohort members had as children. And that's defined in a very crude way. It's whether their father had a manual versus non-manual occupation. So even with this very, very crude index of childhood socioeconomic status, you start to see differences in premature mortality very early. So what many people would still consider young adulthood, and the curve spread out a little bit when people get to their mid-50s. And what you see here is about a twofold difference in risk for premature mortality that persists when you adjust models for all the other predictors of premature mortality that are traditionally used. So this is net of race and ethnicity, smoking, adiposity, so traditional gender, lifestyle, demographic predictors of mortality. You might argue that, okay, the relative difference of twofold is impressive, but the absolute effect here is pretty trivial. We're talking about 3% of those who come from more advantaged backgrounds as children dying versus 6%. But remember, this cohort has only been followed out until their mid-50s at this point. When we follow people into the later years of life, when the base rates of mortality are higher, you see the same effects persist. And they're usually somewhere between 1.5 and twofold, even with, again, these very crude measures of childhood socioeconomic status. There's some evidence coming out now to suggest that these effects might even get passed on to the next generation. So here's some data on pregnancy outcomes from a study we've been working on. This is a study of about 750 pregnant women from across the country whom we interviewed about childhood disadvantage, categorized in a multidimensional way, and we looked at pregnancy outcomes. You can see that the women who had the most childhood disadvantage were most likely to deliver babies preterm, so before 37 weeks, deliver babies that were small for gestational age, and their babies were likely to stay in the hospital about 2.5 times longer after birth compared to mothers who came from less disadvantaged backgrounds. All these figures, again, are net of demographic and obstetric risk factors for these outcomes. Even the mother's current socioeconomic status as indexed by education and income, suggesting that above and beyond those factors, childhood socioeconomic status might be leaving some residue that shows up, as I said earlier, in premature mortality, heart disease, chronic diseases of aging, and in the way that their offspring begin life. And this is important because these outcomes themselves are probably markers for differences in the in utero environment, which there's lots of animal and human evidence to suggest are going to play a role to some degree in influencing risk for the life course health patterns of those offspring themselves. So these might be some indications of transgenerational effects. We'll see. So why should you care about this? This group probably knows all the answers to this question, but what I usually tell people is it has clear implications for families, for public health, for the economy. If we look at the latest poverty statistics, about 16 to 18% of U.S. kids are born into families below the poverty line, depending on how that's defined. Another 20 to 25% are low-income families, so 2x the poverty line. So nearly half of American children are living in low-income families. If the effects we're talking about here are truly effects, and by that I mean causal, then we're going to be feeling the repercussions of this phenomenon for a long time to come, right? If today's kids are at increased risk of, in 60 or 70 years, having strokes and heart attacks, then that's going to be a multi-generational problem that our country is going to be feeling in many, many years. I think from a purely kind of academic or scholarship perspective, these findings also raise some quite interesting questions. So they raise two ones that are particularly challenging for the kind of work that we do. So first is how you take something that is nebulous, like socioeconomic status, and get it inside the body at the level of signaling pathways in cells, and understand how that gets you to differential disease risk. So we need to solve what are thought of as classic kind of mind-body problems. And there's been a lot of traction on that in the past couple of decades, thanks to work done in psychobiology and stress physiology. And we have a reasonably good handle on how you take aspects of the social environment and the physical environment, which tend to co-vary with socioeconomic status, and how those work through the brain, through the HP axis, through the autonomic nervous system to affect the immune system, cardiovascular system, the lungs. So we have decent models and some understanding based on animal work of how that could happen mechanistically. The details are still being filled in, but we've got some of the handle. But besides this kind of classic mind-body problem, there's also this temporal problem that mind-body people aren't accustomed to dealing with. And the temporal problem is that the exposure we're talking about here, low socioeconomic status in childhood, is 60, 70 years before the outcomes we're typically talking about in this research, which is having a stroke or heart attack. And stress researchers are pretty good at understanding the here and now physiology of stress. So Cam or John could tell you a lot about what's happening to hormones in my body as I'm standing up here talking. But what we don't really have a good explanation for is how a stressor that happens today might come back to affect my disease risk 40, 50 years from now. And bridging that incubation gap is a real conceptual and mechanistic challenge for stress researchers. And then the other question, of course, is one about understanding risk and resilience. Not all kids who grow up in challenging circumstances go on to have worse health outcomes. Edith will talk a lot more about this. So we not only need to explain the risk side of the portfolio, but also the resilience side, and hopefully leverage that knowledge of who's thriving despite challenging childhoods to perhaps develop interventions and policies that can make that more widespread. That's the second half of the talk. So before I get to what we think some of the explanations are, let me go through two kind of intuitive and common observations that people often make upon seeing relationships between childhood socioeconomic status and later health outcomes. So the first is the kind of classic, this is really all about bad genes explanation, right? So the argument goes something like this. There is a set of risky genes that tend to predispose families towards lousy health outcomes, towards not doing so well in school, towards getting bad jobs, to maybe not being so smart, to taking lots of risks. All these genes cluster together, and they also tend to cluster together in families. So what you're seeing here is not so much a causal association between childhood socioeconomic status and later health, but instead some kind of familial transmission of a risky genetic risk. So this is a tough criticism to deal with because nobody's been able to specify what that genetic risk looks like. If we knew what the risky alleles were, we could measure them in a GWAS, and we could stratify or adjust for them and find out if that's true, but nobody really has been able to specify it. So you can sort of do two things in these circumstances. One is you can say, all right, well then let's take a look at administrative data from Northern Europe, and there are a handful of studies that have been able to say what happens to the health of kids who get adopted into families of a different socioeconomic status than their biological parents. And by linking those early adoption records to later hospitalization and mortality records, what you see is that the SES of the biological parents and the SES of the adoptive parents independently predict to later health outcomes, suggesting that genes are part of the story, but they're probably not all of the story. You can also look at animal research, and I think this is pretty telling. So one can randomly assign animals to challenging early life circumstances. One can also quibble with how well that represents the human condition of low socioeconomic status. But when we do that, not we, larger we, when other people do that, there is quite compelling evidence that early life stressors, particularly around maternal separation, increase animals' vulnerability to the kinds of problems with their hearts and their lungs that we see in human studies. And I find those studies fairly compelling evidence that this can happen causally. It doesn't mean that it necessarily does in humans, but it can. So to me, the totality of the evidence here is consistent with a causal interpretation of the human data, although there are still gaps to fill in there. And I think there's room for arguing about whether we're there yet. The second explanation that people often arrive at when they see these data are that they're really all about adult SES, right? So the idea that childhood SES and late life health outcomes are related is really more about the fact that in many countries, especially in the United States, socioeconomic status is unfortunately stable across the lifespan, and that kids who grow up in disadvantaged families tend to become disadvantaged adults themselves. And it's that disadvantage when you're in your 50s or 60s that gets in the way of you having long-term health outcomes that are positive. It's more proximal in time, it's more proximal to the disease. There's definitely some of that going on in these associations. Most of the studies in the field adjust for adult socioeconomic status. They do that in ways that vary in terms of quality, but the best studies typically find that childhood SES predicts above and beyond adult SES, suggesting there's some incremental variance. There are also studies that look at this in a slightly different way. So these are data from a cohort that comes out of Johns Hopkins Medical School. It's called the Precursor Study, and this study enrolled a couple of thousand male physicians who graduated from Hopkins in the 50s and 60s when they only admitted males, and they followed them to look at health outcomes over the long term. We're looking at coronary heart disease morbidity here, and you might not be able to see this slide because it didn't reproduce very well, but what it shows is that rates of coronary heart disease in these physicians differ according to the physician's childhood socioeconomic status, and they favor those who come from more privileged backgrounds, again, indexed in a very crude way. Did dad have a manual versus non-manual occupation? So what you see in slides like this is even in people who have made it to the pinnacle of American SES, right? They're physicians, they're highly paid, lots of education, one of the best medical schools in the United States. There is still this residual association between their childhood socioeconomic status and their risk for heart disease, and they've shown this with other outcomes too. So again, consistent with the conclusion that this isn't just about childhood SES being a proxy for education, income, occupational prestige, things like that, that people accrue across the lifespan.
Question?
Yes?
Isn't it a little worrying, though, if even at very high levels of status, you're getting this prediction? Because you would think that, well, here, maybe the differences in childhood environment are not that important anymore, right? I mean, that if these are people going on to be physicians, then maybe there are fewer books to read in one household than the other. But in terms of these kind of gross stressors, maybe you might not expect the childhood environment to vary that much.
Right. The adult environment is pretty powerful here, right? So it suggests that the childhood effects, I mean, to me, this finding suggests that the childhood environment is probably even more important than we initially would have given it credit for.
But my point was that you're talking about this in terms of stressors that are appearing in childhood. But what I'm saying is if at the top 10% of the status distribution, it's still the case that the people at the 1% are doing better than those with parents at the 20th percent down, then you worry a little bit that this is not necessarily about these childhood stressors, but about some other factor, because you're not at the very bottom of the distribution of homes in terms of stresses and stuff like that. You're actually well up the distribution.
Right, so this hinges to some extent on whether you think these are threshold effects that occur because below some level of deprivation, people are really missing out on material things, or you think that they tend to be more gradient-like effects that occur across the distribution. So if one looks at the larger literature on social disparities in health, particularly in adults, we see patterns that are much more gradient-like than they are threshold-like. And that's been a real struggle to explain in the broader social disparity. So why is it that, somebody who's upper middle class has worse health outcomes than somebody who is upper class, whatever you want to define it. But why should small differences in education or small differences in affluence, wealth, income really matter for health? Because clearly everybody in those strata are above the threshold where they have adequate housing, adequate nutrition, adequate access to health care. And the argument tends to be that at those levels, and this is most clear in the Whitehall studies done in the UK by Marmot and his group, is at that level you really are probably talking more about things like ability to control outcomes on a day-to-day basis, ability to manage and juggle stressors, and there's a whole literature on also subjective status, way apart from objective status and material resources, people's perceived positions within hierarchies. And there's some evidence to suggest that those, especially at higher levels of status, that perceived position within the hierarchy, which doesn't necessarily line up with what you think of as objective indicators of status, is an important incremental predictor of outcomes. So we don't know this as well in the literature on childhood SES because almost all of the data come from either administrative studies or much, much older birth cohort studies that have just very crude markers of parental education or parental occupation. And we can't really look at gradients in those sorts of studies. With that said, there are an emerging number of developmental studies that are looking at children and adolescents and young adults that have much more embellished measures of childhood socioeconomic status. And in those places, we tend to see gradients too, suggesting that this may be a really linear thing. But we don't know how that links up to real health problems at the other end of the lifespan. But we can show a linear relationship between early risk factors. So, let me see. All right, so if it's not just about genetics, and it's not just about the accumulation of wealth and education across the lifespan, varying as a function of your early circumstances, then what is it? And we have struggled a lot with this question, as have other groups. And our thinking is that, there's no magic silver bullet, right? There's no one mechanism. It's not just going to be about telomeres, or it's not just going to be about methylation of glucocorticoid receptor, or it's not just going to be about smoking, or it's not just going to be about cortisol. That socioeconomic status is complex. There are lots of dimensions. There are lots of elements to it. Health is similarly complex. People are probably going to get from A to B through multiple different routes. And our models and, more importantly, our empirical work need to do a better job of reflecting that. So here is our, feeble and crude attempt to start thinking about, what kinds of boxes does one want to have in one's mechanistic models. And our work kind of starts with the assumption that in childhood, disadvantage is associated with a whole set of social and physical exposures, and an increased likelihood of those being related to the way that kids' brains develop, to the way their personalities and lifestyles develop, to the way that they engage with the social environment, and that there are biological implications of those things. And our arguments have been that we can probably, based on the animal literature and some of the human literature on caregiving and the HPA axis, the kind of stuff that Cam did as a graduate student, are consistent with the idea that we can probably expect some classic Barker-like programming effects, that during sensitive and perhaps critical periods of development, experiences with stress, experiences with caregiving may get wired into certain physiological systems in ways that are fairly stable across the lifespan and will alter vulnerability to later diseases. But we and others certainly aren't convinced that this is all about a deterministic kind of programming effect. Clearly, what happens after birth or after the early years in terms of the way people's personality develop, in terms of their health behaviors, their lifestyle, in terms of their interactions with their social world back and forth, all of those things probably continue to matter for people's health as they go across the lifespan. And we know from decades and decades and decades of research in human development that early experiences matter for the way people's lifestyles and personalities and social interactions shape up. So our hypotheses have been around both programming effects and what we call accentuating effects or moderating effects. We have focused probably too exclusively on the role that the immune system is likely to play in these health outcomes. And so much of our work focuses on relationships between disadvantage in childhood and how the parts of the immune system that are involved in inflammation get set up and operate across the lifespan. And we focus on inflammation because it helps us conceptually explain how one could get, with the similar early life exposure, to lots of different chronic conditions associated with aging. So for those of you who haven't been following this over the past couple of decades, there's been an explosion of evidence and knowledge that non-resolving inflammation, this kind of low-grade chronic inflammation, the dysthymia of the immune system is the way you can think about it, is associated with risk and plays a causal role in diseases that range from classic autoimmune problems associated with aging, like arthritis, to inflammatory bowel disease, to heart disease and stroke, to cancer, to MS. And then in most chronic diseases, inflammation plays a, if not the, pivotal role in causing the disease to progress and to manifest clinically in a way that interferes with people's quality of life. So our focus on inflammation has been mainly as a way to get to lots of different pathways. And we've made, in a very kind of simplified way, the argument that childhood circumstances are going to affect the way that the cells that orchestrate inflammation behave. And these cells, which are called monocytes in their immature form, and macrophages, once they mature, have a lot of phenotypic plasticity. That's one of their defining characteristics, is that their behavior conforms really well to the environment that they find themselves in. And our argument in a very simplistic way is that kids who grow up in disadvantaged circumstances are, for various mechanistic reasons that I can go into more detail about later if you want, or you can read the papers, are going to be more responsive to threats. And those threats come in the form of pathogens, and they come in the form of tissue damage. And that when these cells see these threats called pathogen-associated molecular patterns or danger-associated molecular patterns, that they're going to launch relatively more aggressive inflammatory responses, that those cells are going to be less sensitive to signals that turn off those inflammatory responses. Those signals mainly come in the form of glucocorticoids. And then as a result of this tendency to have more aggressive responses and be less sensitive to inhibitory signals, disadvantaged kids are going to have more inflammation happening both in childhood and across the life course. And we go on to articulate a variety of reasons that these effects would initially get set up and then why they'd get sustained. And let me give you a very quick primer on inflammation before I get to some data so that it's clear what I'm talking about. So this is a slide of a monocyte, and in its vicinity is gram-negative bacteria. So what happens here is this molecule that sits on the surface of these gram-negative bacteria, this red dot here, is called LPS, lipopolysaccharide. And when bacteria are in their vicinity, monocytes sense the presence of these bacteria through this molecule LPS. And LPS binds to the toll-like receptor that sits on the top surface of these cells. And that binding event triggers a signaling cascade. And you don't need to remember the details of any of this, except for right here. The big point is that when that signaling cascade gets launched by LPS binding to TLR4, we get activation of this molecule right here called nuclear factor kappa B. And nuclear factor kappa B is what's called the transcription factor. And it's called that because when it gets activated, it goes into the nucleus and it initiates transcription or gene expression, right? So nuclear factor kappa B scoots in, switches on genes, and those genes get transcribed into RNA, and the RNA eventually gets translated into proteins, and those proteins are called cytokines. They're messengers that immune cells and other tissues in the body send out when there's been infection, when there's been injury. And these cytokines are what orchestrate the inflammatory response. They help immune cells to get rid of the bacteria or the virus, if it's a virus, to heal the tissue that's been damaged, and to resolve the problem. So a lot of what we're going to do in these studies is we're going to take people's immune cells and we're going to stimulate them in a test tube with this molecule LPS, and we're going to look at how much of these cytokines they produce, and we're going to look at some of these molecular pathways to see how they're affected. So how do you stop inflammation or slow it down once you get started? As I said, cortisol is one of the main anti-inflammatory signals. So this is why if you've got a rash and you go to the pharmacy and ask the pharmacist for what you should use, she or he will typically give you some kind of synthetic version of cortisol, and that's because it blocks the inflammatory response. Oops, I shut it down. All right. So, what does cortisol do? Cortisol crosses the cell membrane. Did you see that? A little quick, I got to slow it down. It crosses the cell membrane, and what it does is bind to these things inside the cell called glucocorticoid receptors. And the glucocorticoid receptor complex ties up nuclear factor kappa B and prevents it from going into the nucleus to initiate the transcription of these genes involved in cytokine production. And that's why cortisol, hydrocortisone, and synthetic versions of it are so effective in treating inflammatory problems, because they tie up this linchpin of the inflammatory response. So, in some of our early work, I did it again. In some of our early work trying to test this hypothesis, our kind of first study in this area was about 10 years ago, and we recruited just a really small sample of healthy young adults who were recruited to either be high or low in childhood socioeconomic status simply according to the occupational prestige of their parents' jobs when they were in childhood. And to prevent childhood and present socioeconomic status from becoming confounded, we matched on a case-by-case basis for the subject's current SES. And all the graphs I'm going to show you are net of these covariates, so some of the common demographic and lifestyle correlates of SES that you might be concerned about as confounds. So the first thing we're going to do is just take this small sample of subjects, draw blood, put the blood into a test tube with various bacterial and viral stimuli that are going to make the cells think that they're facing an infectious threat. And when we do that, what we see is that the cells of those who come from more disadvantaged backgrounds produce more of this classic inflammatory cytokine called interleukin-6 than the cells of subjects who come from more privileged backgrounds. Again, this is net of that set of demographic and lifestyle covariates I told you about. It should be net of their current SES. We see this with lots of different kinds of activators. So this in particular is the kind of stimulus that would make the cells think they're seeing a double-stranded RNA virus. This is a stimulus that makes them think they're seeing bacteria. We don't see it with every different thing that activates these monocytes and macrophages, but we see it with more than not. We see it with most of the classic pro-inflammatory cytokines, so this is IL-6. We also see it with IL-1 and TNF-alpha. It's a pretty consistent finding when we challenge these cells called monocytes with stimuli, that those who come from more disadvantaged backgrounds across the lifespan tend to show larger responses, so a greater inflammatory response, at least in vitro. Going the wrong direction. So we can also take these cells and try to understand, all right, so if there is a larger response to these microbial threats, why is that? What are the signaling pathways in the cell that are relatively turned on or turned off by early experience? And one of the ways we can do that is through transcriptional profiling. So this is a technology where we take the RNA out of these cells, and we can use the RNA that we've extracted to measure the expression of all of the different genes in the human genome. So this is about 25,000 different transcripts. You got a lot of dependent variables in this equation. You've understandably got issues with type 1 error, false discovery. There are a lot of methods worked out to contend with that stuff, which you can talk about later if you want. When we do analyses that deal with all those issues fairly efficiently, and we go back and do some high-level bioinformatics, the theme of what we see is that again, these are young adults, that in young adults who grew up in more disadvantaged circumstances, there is excess activation of genes that are regulated by nuclear factor kappa B. Now, if you remember back to that cartoon that I showed you a few slides ago, nuclear factor kappa B is the linchpin of that inflammatory signaling cascade. It's the thing that slips into the nucleus and turns on the genes associated with inflammation. So even when we have an activated people's cells, if we just take out the cells of subjects who come from more or less disadvantaged backgrounds, and those from more disadvantaged backgrounds, their cells are already making more NF-kappa B products. They're already expressing this gene and the signaling pathway at higher levels. At the same time, they show less activity of genes that are regulated by glucocorticoids, right? And if you remember, again, back to those slides I showed you, this is the on switch for inflammation in these monocytes and macrophages, and this is one of the off switches, right? So in the folks who come from more disadvantaged backgrounds, we see excess activity in the on switch and decreased activity in the off switch. So this is consistent with some of the hypotheses that I showed you earlier on. How early do we see this? Here's some data from a collaboration we had in Manitoba. So this is a birth cohort of kids whose SES was tracked from the time they were born until they were age 9. And here we're using house size as a proxy for family socioeconomic status, which in Manitoba is a fairly reasonable proxy. Most people have their mortgages paid off by the time they're in their 40s or 50s. You can get bigger houses. And we wanted to look at a measure that was a little more dynamic. And what you see is that, in our sample of a little over 300, you can cluster people into groups based on the size of their house. There's a group of subjects who have small 2-bedroom houses, small for Manitoba, a group who has larger 5-bedroom houses, and everything in between. And then you've got this one group who's got what you might think of as social mobility, right? Their house size increases when the kids hit about kindergarten age. So we tracked this prospectively, and then at age 9 we collected blood from the kids, and we stimulated their blood with LPS and looked at cytokine production, IL-6, the same thing I showed you before. And what we see here is an interesting thing that looks pretty much like a gradient up to here, where the greatest production of this inflammatory molecule when the cells are challenged with bacteria is in the kids whose houses are the smallest in a stable way. The least production is those who come from the biggest houses. You see in the group whose house size increased across time, levels are fairly equivalent to those who had stably large houses or stably high SES, if you're willing to agree that this is a reasonable marker of family SES. This is consistent with the possibility that it's a recency effect as opposed to an early life effect, and that where you are at age 9 matters. We are trying to replicate those findings in different ways and look at the kids as they get older now. There's also some interesting into the next generation findings coming out. So I showed you those findings about pregnancy outcomes. We've been looking at similar issues in the context of childhood asthma, where we do a lot of research that we're unfortunately not going to get to talk about much today. But these are kind of hot off the press findings from a study that we've been running for the past few years. And these are looking at kids between the ages of 8 and 15 with active asthma, who we study pretty intensively. We study both clinical outcomes and biological pathways that might explain them. And so what we're doing here is taking a couple hundred kids with asthma, drawing blood, activating their cells in a test tube with stimuli that cause asthma to get worse, right? And we're looking at the production of signaling molecules that cause gunk to accumulate in the lungs and interfere with these kids' quality of life. And what we see here is that production of these cytokines that are important in asthma pathogenesis vary according to the childhood SES of the kid's parent. And again, this is net of the family's current SES, wealth and education, and it's net of other demographic and lifestyle covariates that you might be concerned about. And what you see here is that if you're a kid with asthma and you come from a family where your parents had lower childhood SES, you're going to produce more of these Th1 cytokines and these Th2 cytokines, all of which contribute to the kinds of inflammation and obstruction of the lung that causes worse clinical outcomes. And consistent with that, if you look at measures of asthma control, so this is the asthma control test. It's a common clinical tool for assessing how under control kids' asthma is, and you ask the kid and you ask the parent, you see in both cases that, again, that varies as a function of the parent's childhood SES net of the current family SES. I am going to stop now. I've gone a little bit longer than I probably should have, and turn it over to Edith. Let me just get her slides up here.
All right. Thanks. Okay. So one way in which we have not yet integrated is in the programs we use to give our talks. So Greg uses this fancy Keynote program that I don't know how to use, so we weren't able to just merge all of our slides
we have a
together.
question about
go
one
ahead.
of your slides. Should we hold it?
Oh, I don't know. How do you want to do that? Do you want
Yeah, why don't you hold it?
stop for questions?
That's fine. Yeah, that's what I was saying.
Well, maybe not. Here we go. Okay. So my portion of the talk is going to be sort of more psychological in nature. And that is that one of the things that we've thought about as we've kind of done some of this work on early life adversity and health is the notion of resilience, or the idea that despite the fact that kids who grow up under a lot of disadvantage are at risk for a variety of poor health outcomes, that there are in fact a minority of individuals who manage to thrive despite having come from very adverse circumstances. So if we look at this, for example, in the educational domain, many of you, I think everyone here at the center is probably very familiar with statistics such as these. So if you look at, for example, the percentage of students who graduate high school who then go on to college, what we see here is sort of a traditional gradient relationship, so such that those kids who come from the highest income quintile of family income are significantly more likely to go to college than those who come from the lowest income quintile. But at the same time, one of the interesting things about this graph is that if you look at this group that you would think of as the most at risk or coming from the most adverse circumstances, those who come from the lowest income quintile, what we see is that 50% of them are going on to college. So that in spite of the adversity that they've dealt with throughout their life, that they've come from, we have a good number of essentially what we could think of as success cases. Kids who are making it to college despite having grown up under adverse circumstances. Now, a lot of this work about thinking about this notion of resilience, so good outcomes despite adversity, has really focused on the academic or the sort of behavioral domain, so things like educational outcomes, things like substance use outcomes, juvenile delinquency kinds of outcomes. And so one of the questions we were interested in is, what about physical health? That's sort of the sort of broad area that we study. What are the potential physical health consequences of resilience in this kind of domain, of something like a kid who comes out of a very difficult neighborhood actually making it to college? What happens to them in terms of physical health? There's very little that's known about this because there's so much focus on getting these kids to college to succeed academically, to stay out of trouble, stay out of jail. But what happens to them in terms of physical health? And as we started to think about this, we started to wonder about whether, in fact, in terms of physical health, there might be some sorts of costs of resilience. So that is that if you think about the type of kid who can make it out of a neighborhood filled with poverty, a lot of adversity, violence that they encounter in their lives, and who can make it into college when nobody in their family has ever been to college, what it takes for a kid like that to succeed is sort of this sort of accumulation of years of sort of hard work, dedication, sort of single-minded determination to kind of beat the odds. And in particular, sort of the idea of high levels of sustained self-regulation to avoid the sort of temptations or distractions in one's environment and to kind of focus and kind of succeed, make your way into college. And so there's this notion, though, that kind of sustained effort over time, sustained efforts at self-regulation, might in fact take some kind of internal physiological toll on these youth. And that if it did, you might see it in terms of markers that indicate kind of cumulative or biological wear and tear. So these are things like allostatic load, which is a kind of multi-system indicator of physiological risk. It's thought to be sort of this kind of cumulative indicator of physiological wear and tear on the system that comes from years of having to sort of achieve homeostasis in the face of kind of ongoing stressors. And this kind of wear and tear is thought to manifest in dysregulation in multiple kind of physiological systems like the cardiovascular, endocrine, and immune system. And so allostatic load is kind of a composite and thought to be kind of an early sign of physiological dysregulation that has been found to be predictive of later disease risk like cardiovascular disease. So we have the notion of things like allostatic load. There's also a notion of, as another biological indicator of wear and tear, epigenetic aging. So this is a measure that's done in immune cells of DNA methylation profiles. And so the idea here is that in using this approach, you can get what's thought to be kind of a biological indicator of aging in cells. And you can compare that to a person's chronological age. So what you're looking at here is the disparity between biological age and chronological age, with the idea that those who are showing signs of physiological wear and tear are going to be higher on epigenetic aging, or showing sort of this sort of more advanced, faster biological aging relative to their chronological age. And again, markers of epigenetic aging are also, a faster epigenetic aging is found in things like tumor cells, has been associated with risk of mortality down the line. So we could think about the idea that if there is maybe a physiological cost to resilience, it might appear in things like allostatic load, epigenetic aging, or perhaps even in clinical kinds of health outcomes. So we looked at this in a number. We've looked at this in a number of studies that have, that we've been working on kind of recently, and in particular in studies of African American youth. So across a variety of different samples that we have of usually samples of several hundred African American adolescents, we measure sort of disadvantage or in terms of indicators of family poverty, so living below the poverty level, receipt of TANF, parent unemployment, as well as indicators of neighborhood poverty level, like census, based on census tract information. And in these youth, so these are kind of longitudinal samples that we have access to, that we collaborate on with a colleague at the University of Georgia, Jean Brody. What happens is that these adolescents were recruited first at between the ages of 11 to 13. During that period, there was a battery of psychological assessments done, one of which is self-control, which is sort of this key factor that we think if these kids are going to succeed, they're going to have to show a lot of self-control over time, but at the same time, that might also have a cost to it. So these measures of self-control are based on teacher ratings of these kids in the classroom, ages 11 to 13, where they're asked about, sort of the extent to which these kids can stick with what they're working on, pay attention in class, set goals, and make progress towards goals. And then what happened was we followed these kids over time. And at the age of 19, we assessed both behavioral outcomes, so things like externalizing behavior, so this is the extent to which they're engaging in aggressive behaviors, kind of rule-breaking behaviors. And then at the age of 19, we also had the opportunity to go back to these youth and draw blood on these youth. And so that allowed us and to collect a variety of physical and hormonal measures to sort of allow us to take this measure of allostatic load. And what we found when we did this assessment and looked at this is that we found some interesting interactions between SES and self-control in predicting externalizing behaviors and allostatic load. So let me walk you through this graph, and then I'm going to show you a few more kind of from different samples that are kind of along these lines. So the way these graphs are set up is level of self-control, so this is measured at age 11 to 13, is on the x-axis. So it's a continuous variable, goes from low to high. The SES variable is a family SES. So again, that's a continuous variable, but for the purposes of graphing the interaction, we've broken it down by low and high. So lower being those families who are living under poverty, higher SES those who are not. On the y-axis are outcome variables. So here in the graph on the left is externalizing problems, there's aggression kinds of behavior problems. On the graph on the right is our physiological indicator of allostatic load. So in both cases, higher numbers indicate worse on those measures, so more externalizing behaviors and a higher level of allostatic load, which is considered worse. So what we see here is that this interaction, so if you focus on the low SES group, which is the dotted lines, what we see here is that among youth who grow up in low SES families, having high levels of self-control is good in terms of behavior problems. So those youth who are higher in self-control are exhibiting fewer behavior problems at age 19. But at the same time, in these same youth, that high level of self-control turns out to be bad in terms of their physiological profiles. So among these, again, lower SES youth, the more they're engaging in self-control, the higher their levels of allostatic load. And these youth are showing the highest allostatic load of any sort of group in this sample. Whereas among the higher SES kids, it's the kind of relationship that we often think of in psychology. Self-control is often thought of as a good variable. You want kids to have a lot of self-control. In higher SES samples, higher levels of self-control are associated with lower allostatic load. So it comes out specifically only in the lower SES youth. We see the same patterns when we look at epigenetic profiles. So this is in another sample that we did, again, a sample of African American youth who are characterized as either coming from more disadvantaged, so lower SES families, or higher SES, less disadvantaged families. We again measured self-control, and then 5 years later took a measure of epigenetic aging. So again, now here this time the sort of outcome variable is plotted down here. Higher scores indicate faster epigenetic aging, more rapid epigenetic aging, which is considered bad. So what you see here again is that among youth who grew up lower in SES, higher levels of self-control are associated with faster epigenetic aging compared to lower levels of self-control, whereas it's the opposite pattern among those youth who come from higher SES circumstances. So self-control is good in terms of epigenetic aging if you come from IRS background, but it is bad if you're coming from a more disadvantaged kind of background. We see a similar kind of profile if we look at kids who actually make it out of poverty, out of impoverished neighborhoods, and make it to college. What happens to these youth in terms of both behavioral outcomes as well as physiological profiles? So again, in a sample of youth, what we looked at here in this case is based on the neighborhoods they came from and the percentage of people living below poverty in their neighborhoods. So that's graphed along the x-axis. And then y-axis here is, again, our sort of behavioral and our physiological outcomes. So in this case, we looked at substance use, rates of substance use, so drug use, alcohol use, smoking, as well as allostatic load. So again, higher scores on both are considered worse. So what we find here is that among those who come from higher SES neighborhoods, so for those who come from neighborhoods where there's very little poverty, it doesn't actually, rates of substance use are pretty uniformly low. But as youth start to come from neighborhoods with higher levels of poverty, it actually matters whether they make it into college or not. So if they make it into college, they have low rates of substance use behaviors compared to if they do not make it to college. So those who come from high poverty neighborhoods who don't end up going to college, they have higher rates of substance use compared to those who come from neighborhoods with higher levels of poverty but who do make it to college. So again, that idea that sort of self-control may be helping these youth to refrain from or to stay away from drug use in the context of high neighborhood poverty. In contrast, when we look at allostatic load, it's an opposite pattern again of sort of the impact of making it to college, such that if you come, a youth who comes from a neighborhood with high poverty rates, that those who make it to college have higher levels of allostatic load compared to those who do not make it to college. So that sort of ability to get into college, while good for you in a lot of different ways, turns out to be associated with more sort of physiological risk in terms of higher levels of allostatic load if you come from disadvantaged neighborhoods to begin with. One last study that we've done that shows this phenomenon, so again across a different variety of samples. So this is a sample we worked together with Sheldon Cohen, who does this kind of interesting cold study paradigm where he actually he brings research participants into his lab and he actually exposes them experimentally to a cold virus. And the idea is to be sort of to do a study in a very controlled way about figuring out who is at risk for getting a cold. So this is kind of a clinical outcome here. But rather than just sort of interviewing people or assessing who gets a cold and who doesn't, and that's completely confounded by did you get exposed to a virus or not, he exposes everybody to the virus, and it turns out that when you get exposed to a cold virus, not everybody who does actually gets the cold clinically. So he's looking at who's at greatest risk to get a cold clinically if you've been exposed to the cold virus. And so what happens is that, so in this case, the outcome here is the proportion of people who develop an upper respiratory infection, URI. So higher numbers is bad. And so it's broken down again by SES and then by a variable comparable to self-control. So childhood SES is coded by parental home ownership. So the number of years that families own their own home. So this would be lower in SES and this would be higher in SES. And in this study, we didn't have a measure of self-control, but the closest thing we had to it is a personality trait called conscientiousness, which is kind of, it encompasses self-control, but also encompasses things like being hardworking, organized, responsible, reliable. So what we find here again is that when you look, and these analyses are done within, again, the African American sample within his cold study. So when you look at this, among those who grew up in lower SES environments, so with families that didn't own their own homes, as conscientiousness increases, your risk of getting a cold increases. Whereas among those who came from higher SES families, as consciousness increases, risks of getting a cold decrease. So what we're finding is that those youth who come out of very difficult neighborhoods or family environments, but who manage to succeed academically and behaviorally, so demonstrate resilience in these regards, may actually be experiencing a physiological cost to the success. But that leaves us with the question of, well then, are there factors that do protect low SES youth from poor health outcomes? So again, a similar kind of graph, this is another one of Sheldon Cohen's study, cold study. So if we look at rates of developing a cold after you've been experimentally exposed to a cold virus, there's a traditional SES gradient such that those who come from lower SES childhood homes are significantly more likely to get a cold compared to those who come from higher SES homes. But again, this point about resilience that I showed earlier with making it to college, going off to college, is true in terms of physical health outcomes as well. So even among the highest risk group, those individuals who come from low childhood SES families, about over 40% of people who are exposed experimentally to a virus do not end up getting a cold. So despite the fact that everybody got exposed to the virus, you still have a good chunk who are showing resilience in a physical health context. So why is that? What are the kinds of factors that might predict who then does well physically in terms of physical health despite growing up in adversity? So there are a number of reasons why this might be. I'm just going to go over one today, so one factor that we've looked at for the sake of time, and that is a notion of nurture and parenting. So this is a factor that's been studied widely in the developmental psychopathology literature. It refers to the idea of understanding, attention, warmth, and affection from a parent. And in the developmental psychopathology literature, this is probably the single most robust factor associated with good outcomes among kids who experience adversity, a lot of adversity in life. So youth who grow up under adversity but who have nurturant parents show better psychological adjustments, so things like fewer symptoms of depression, fewer behavioral problems, better social competence. But the vast majority, again, of this literature has focused on that sort of behavioral and social emotional functioning kinds of domains. And there's far less, again, known about whether nurture parenting would be beneficial in terms of physical health. Now, from the animal literature, we know that if you accept that the potential equivalent of nurture and parenting in an animal model, so that is high levels of licking and grooming from mothers in rat pups, we know that experimentally has been shown to buffer elevated physiological responses to stress when those rats then later grow up. But there isn't a lot of data in humans. So we looked at this in a large national sample called MIDAS. It's a midlife in the U.S. study, in which we had measures of the childhood socioeconomic circumstances that participants grew up in, so as based on their parents' level of educational attainment. We had a measure of parental nurturance, so again, sort of measures of the sort of warmth, attention, and understanding they received, and it was asked separately about both their mothers and their fathers. And here we were able to measure an outcome called metabolic syndrome. So this is, again, this is a little bit similar to allostatic load, a cluster of risk factors that are known to be thought to be early precursors to diabetes and cardiovascular disease. So this is kind of elevated levels of adiposity or obesity, elevated blood pressure, high levels of cholesterol, and impaired glucose control. So what we do is there are International Diabetes Federation cutoffs, clinical cutoffs for being called elevated on each of these dimensions, and we count up the number of symptoms that a participant is elevated, clinically elevated on across these different measures. And what we find here is that there are significant buffering effects of maternal nurturance. So the way this graph is laid out, on the x-axis we have their childhood socioeconomic status, so parent educational attainment, ranging from less than high school to high school degree, some college bachelor's degree or higher. On the y-axis is the number of metabolic syndrome components. So these are the numbers of components on which they show clinically elevated signs of, so higher is bad. And we see the graph, it's graphed at 3 different levels of maternal nurturance. So what we see here is that for those participants who grew up under higher SES circumstances, the extent to which your mom, you remember your mom being warm or not, nurturant or not, doesn't actually matter at all in terms of your metabolic symptom profile. But if you grew up in a lower SES household, then maternal nurturance mattered. And it mattered such that those who grew up in higher those who remembered experiencing higher levels of maternal nurturance have significantly lower levels of metabolic symptoms compared to those who grew up similarly low in SES but who reported not having a nurturant mom. And among those who grew up low in SES but had a nurturant mom, their metabolic symptom profile is actually more similar to the higher SES, those who grew up under higher SES circumstances, than they are to their low SES counterparts. Now, I should also note that these findings come out, interestingly, only for maternal nurturance, not for paternal nurturance. So paternal nurturance had no main effect or interaction effects predicting metabolic symptoms. So it was only the extent to which you were called your mom is nurturing and not so much your father, unfortunately. Although I think there are explanations for that I can go into as well. So for the sake of time, I'm going to skip this next one and just point out that so now that these observations, we've done a series of studies on the kind of role of maternal nurturance and found some of these buffering effects fairly consistently. We think they're really interesting findings, but again, they're all observational, and so we can't make any claims about causality and whether we know if maternal nurturance would play a causal role in buffering kids who come from low SES circumstances. So one of the questions this led us to think about, though, is, given these intriguing findings, is would it be possible to change people's parenting styles? And if you could change parenting styles, could you make an impact then on some of these physiological outcomes? And so this is the last study I'm going to present today. So that is where we had an opportunity to partner with again, Jean Brody at the University of Georgia on a parenting intervention study. So this is, again, a sample of African-American families, mothers with their 11-year-old child. So in this study, families were randomly assigned to either a parenting intervention known as SAFE, Strong African American Families, or to a control group. If they were assigned to the parenting intervention, parents were provided with sort of instruction and lessons around sort of nurturant parenting techniques, so the idea of providing emotional support, listening without criticizing, but at the same time sort of parenting that emphasized high expectations and good communication. Youth were sort of talked to about in terms of the importance of obeying household rules and strategies for coping with the kinds of stressors they had in their environment. And so what happened was, if they were assigned to the intervention group, there were 7 group sessions over the course of 7 weeks. Each session lasted 2 hours. So the first hour, the parents and the kids were separated and given separate lessons. Second hour brought together and so that they could practice some of the strategies they were taught. If they were assigned to the control group, they basically received 3 mailings about general child development and general health, but nothing about parenting specifically. So what happened was, this parenting intervention was done. It was designed actually initially to try and reduce things like delinquency behavior, substance use behavior in kids. And it was found to be efficacious for that. But 8 years later, after this intervention was done, we had the opportunity to go back to these youth and draw blood samples on these youth. And what that allowed us to do was to measure these kinds of pro-inflammatory cytokines that Greg talked about, to say, would there have been potentially any effect of having gone through this intervention versus not, in terms of these pro-inflammatory cytokines like IL-1 beta, IL-6, and TNF-alpha, measured 8 years later. And what we found was that we measured 6 different cytokines across all 6 6 cytokines, we found a consistent pattern such that those youth who were assigned the parenting intervention had significantly lower levels of these pro-inflammatory cytokines compared to youth who did not go through the parenting intervention. This was true for interleukin 6, it was true for IL-1 beta, it was true across all 6 of those cytokines that we measured. So again, some evidence here that in a causal way, maternal warmth or nurturing parenting can help to buffer those who grow up under adversity from metabolic symptoms and heightened inflammatory profiles. Okay, so let me stop there because I know we've gone a little bit long, and that way we can take questions to either of us. And thank you for your